Hello everyone, I hope you all are doing well. So in this video, I'm going to share my MSc Life Sciences interview experience at IISc Bangalore. So my name is Shubham Prakash Dutt Mungai and I have secured All India rank 24 in IIT Jam Biotechnology, All India rank 129 in GATE Life Sciences, and I was amongst the top 123 candidates who were selected for TIFR Mumbai interview. I have been selected for MSc program at IISc Bangalore and IIT Bombay, and I have also been selected for integrated PhD program at IISc Bangalore and IISc Pune. So let's begin with my MSc interview experience first at IISc Bangalore. Rest I will upload in a series. So my interview was on 18th May in the morning shift, and in I was in panel second, and in my panel there were four people, so or four professors there were there, and the interviews were being conducted in a classroom only. So my interview started like first my name was called at around 12 p.m. because I was second last or maybe third last in my panel. So my name was called at around 12 p.m. and then they asked me to sit and they asked me about how am I and then if I'm doing well. So then my interview starts. They ask my for my introduction first. So I just told my basic introduction, what is my name is and where am I from, what I'm currently pursuing. Then they asked me about my topic of interest, three topic of interest. So the first thing that you have to keep in mind in any interview, you should be prepared with any three topics of interest. So I said that I like biochemistry, molecular biology, and genetics, and these are my topic of interest. So mostly what they do is they basically try to ask you from your topic of interest only. Sometimes like some few questions would be there that they may ask from any other topics. In my case, I had been asked from cell biology in this interview. So I'll share how my interview was. So first, there was a professor who was sitting behind the three, like three were sitting in the first row, and one professor was sitting in the back row. So he started the interview and he asked me that, do you like protein biochemistry? So I said, yes, sir, I'm interested in protein biochemistry. So he asked me, how would you, like his first question was, how would you determine, like if I gave you a protein sample, so how would you determine if the protein is a membrane protein or it is a cytosolic protein? So I answered that, sir, if I have to determine that whether the protein given is a membrane or cytosolic protein, what I can do is I can sequence the protein and usually membrane proteins, for example, membrane proteins which are crossing the membranes, so they have a transmembrane domain. So this domain is of approximately 20 to 25 amino acids and it contains of hydrophobic amino acids in chain. So I would sequence the protein first and then I would determine whether I can find this transmembrane domain or not. So what I can do is I can plot a hydropathy plot for my protein 2, and after plotting the hydropathy plot I can determine whether whether the given protein in the sample is cytosolic protein or it is a membrane protein. For example, if it is a membrane protein, then I will see a peak in the hydropathy plot. So then his immediate question was like, do you know what hydropathy plot is? So I said, yes, sir, I know what hydropathy plot is. So he asked me then, please draw your hydropathy plot. So I just drew a simple hydropathy plot. You can see in any book or you can Google it too. So I just drew a simple hydropathy plot and just I showed peaks and dips. So I then explained that, sir, the peaks are usually indicate that there is a series of hydrophobic amino acids in the protein, and since if the peak is of approximately 15 to 20 amino acids, I can determine that it is a transmembrane domain. It is spanning the membrane region. So he said like, yeah, very good. Now his next question was like, do you know what are the types of what are the types of hydropathy plots? So I said, no, sir, I have not studied hydropathy plots in that detail. See, if you don't know anything, like if you don't know any information, you can simply state that, sorry, sir, I have not read about this, because they are not like there to test your like kitna tum ratta maar sakte ho, how much can you mug up. If you don't know, you can simply say, sorry, sir, I don't know. they won't mind for two few questions, like two three questions. They don't mind about it. So he asked me like, do you know hydropathy? What type of hydropathy plots? He said some name, but I said sorry, sir, I have not read in that detail. Then his next question was, so do you know like types of amino acids? Now the in the context we were talking about, so I said yes, sir. In our context, there are two types of amino acids. There could be hydrophobic amino acids and there are hydrophilic amino acids. So he asked me, can you name some hydrophobic amino acids for me? So I said, yes, sir, I can surely do. Then I started naming glycine, proline, alanine, phenylalanine, tryptophan. So he was like, okay, okay, it's fine. Now his next question was, if do you think that the amino acids like the hydrophobic amino acids have same hydrophobicity? All the hydrophobic amino acids have same hydrophobicity. So do you think that? So I said, no, sir, I don't think so that all hydrophobic amino acids have same hydrophobicity. They have different hydrophobicity. So he asked me, so how would you determine their hydrophobicity? Like, how would you rank them? If you were to rank how much hydrophobic an amino acid is, so how would you rank amino acids on the basis of their hydrophobicity? So now, if you see this question, like it is a question where you don't know, maybe you can know, but I didn't know what is the ranking of hydrophobicity of the amino acids. So in this case, what I did was like I processed some information in my mind and then I answered. For example, like I answered in the context of chemistry. So I said that, sir, if I were to rank these amino acids based on their hydrophobicity, I would rank phenylalanine and tryptophan the highest. Why? Because phenylalanine and tryptophan both contain a, a benzene ring, and this benzene ring will provide them maximum hydrophobicity compared to other amino acids. Now, his immediate question was, what about tyrosine? Now, this is what actually I was thinking. Like he would ask me this, so I said, sir, tyrosine would be ranked a little lower. Actually, it would be ranked lower than maybe most of the hydrophobic amino acids because tyrosine also contain a hydroxyl group attached to the benzene ring. So it is actually a phenyl, phenol group. So I said that, sir, then since it contains a hydroxyl group, it can also act as a hydrophilic amino acid. So his reaction was, yeah, very good. So then he asked me, okay, now continue your series. So I said, then sir, I would simply rank hydrophobic amino acids based on their carbon chain length. For example, if any amino acid's carbon chain length is higher, then I would rank them higher, and then I would decrease the rank according to their carbon chain length. So he was like, okay, yeah, very good. Now, his next question was a bit tricky. Actually, I was not able to figure it out yet. I'm actually still trying to figure out the answer to this question. So, his next question was, suppose I have given you a protein, you do not know its amino acid sequence, and you do not know what R group is at each position. So I have given you a 3D structure of the protein. It is a tertiary structure, and I have plotted the structure for you in a 3D graph with axis X, Y, and Z. So his question was, if I were to randomly mark a point x1, y1, z1 on the graph, and you do not know about the R group of the amino acids of any amino acid. You just know about the peptide backbone. So you have only two details, that you know the structure and you know about the peptide backbone. Like you know how the peptide backbone is. So how would you determine that the point which is marked by me have a have an exposed R chain, R group, or a collapsed R group? So he basically wanted to ask that how would I determine that at the point the amino acid is hydrophobic or hydrophilic? Now this question was a bit tricky for me. First, I stated that now see, it is not necessary that you know everything. Sometimes these professors, what they do is they try to push your limit. They want to see how much you can think. So it is not necessary that you can answer everything, but yes, you should always tell what your thought process is. So I definitely did not know how to process this question, and I was not able to process it at that time. So first answered, like I started answering in a way that, sir, if I don't know the R group, I cannot determine if the amino acids is hydrophobic or it is hydrophilic. So he said, but I've given you a problem, and I've also said that you know the peptide backbone. So can you think of any way? So I said two, three things, but at the end I was not able to answer it. So I simply stated, sorry, sir, I'm not able to process it right now. So it is like that they won't like reject you if you do not know anything. If you don't know about it, let it be. Just say what your thought process is. So I said that, sir, maybe I can check the dihedral angles, the psi angles. So he was like, okay, it's fine. Then at the end, like I just said that, sorry, sir, I'm not able to process it. So he was like, it's fine. Then you were halfway there. So then there was a second professor. So he asked me that, Shubham, let's design an experiment. So I said, okay, say fine. So he asked me that, let's suppose there is a protein, and it is we are like recently sequenced this protein. We do not know much about it. So I'm giving you a task that you have to determine in which organelle this protein is present. So how would you do that? So now he asked me to take him in stepwise manner. Like I would should say all the steps. How would I do it? So first I thought, like, how can someone ask this simple question? So I just said that, sir, I would do fluorescence tagging, and then I will simply see in which organ it is getting transported to and done. So he was like, no, 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 beta, this is not how we do it. Like it's okay that you have fluorescently tagged, but how would you determine at which cell organelle it is getting transported to? So then I thought for some time, and first, I was not able to get it, but yes, then I figured out what he was trying to ask. So then I said, like, sir, can I change my approach? So he said, yes, yes, you can change, you can try any approach. So I said, sir, maybe what I can do is first I just like it is like slipped out of my mouth that let's assume that the protein is getting transported to mitochondria. So he said, yes, yes, you can assume that the protein is getting transported to mitochondria. So I said, sir, if it is getting transported to mitochondria, so maybe I can check the sequence of proteins which is already present in the mitochondria, and I can compare it with the sequence of our protein, and then we can maybe find some homology sequence. So he was like, okay, it is fine, but do you think that it is necessary that these proteins have homology sequence, like all proteins in mitochondria have some homology sequence? So I said, no, sir, it is not necessary. Then he asked me, so how would you do that? So then I came back to my last approach that I said that, sir, let us assume that the protein is in mitochondria. So maybe what I can do is after fluorescently tagging the protein, I can do centrifugation. So he asked me what kind of centrifugation. So I just said that density gradient centrifugation. He was like, okay, okay. Okay, and then continue. So I said, sir, I can perform density gradient centrifugation, and then I can separate out cell organelle components, like I can separate out mitochondria, and then simply it is done. So his question was like, but how would you determine that the thing that you have separated, like if you are saying that you have separated mitochondria, so how would you say that the thing that you are looking at, you have separated is mitochondria? So I thought for 5 to 10 seconds, and suddenly it clicked to me that marker enzyme, succinate dehydrogenase. So I just said, sir, I can simply test it for marker enzyme, succinate dehydrogenase, and then I can confirm that the organelle which I'm looking at is mitochondria. And if I get fluorescence, that I can simply determine that yes, it is the protein is present in mitochondria. So he was like, yeah, very good. Then he simply asked me some like theoretical question, like what, how would you do if you, like if it was a plant cell and and the protein was getting transported to chloroplast. So he was just wanted to ask, okay, if I know the marker enzyme of chloroplast. So I said, yes, sir, I can test it for Rubisco, and then using this same step, I can determine. So he was like, yeah, very good. Now his next question was, let's suppose, do you know how this, like this first question was, how do you know how these proteins are transported to the cell organelles, their respective cell organelles? So I said, yes, sir, this comes from the concept of protein trafficking, that the protein has usually has a signal peptide, which is usually attached to N terminus or C terminus of the protein, and then using the signal peptide, the protein gets transported to the respective cell organelle. So his next question was, so how would you determine on where the protein's signal peptide is? Like for example, using the same protein which is present in mitochondria, which we just discovered, how would you determine where its signal peptide is located? So then I said that, sir, we are assuming, since we are assuming that the signal peptide is either present on N or C terminus. So what I can do is I can set up two experiments. Like I will the experiment we just performed, I would take it as control. So we know that the protein is present in mitochondria. Now I will set up two experiments. In first experiment, I will delete the N terminus of the protein, and in the second experiment, I will delete the C terminus of the protein. Now, what I will do is, I will let, after deleting the N and C terminus, I will let the protein fold normally, and using the same steps, I will again check that after the protein, like after the protein is translated, and after I deleted the N and C terminus in the different setups, so I would check whether I am still able to find fluorescence or not. For example, after deleting the N terminus, if I find fluorescence in the protein, sorry, in the mitochondria, if I find the fluorescence in mitochondria, then I can confirm, yeah, that the signal peptide is attached to the C terminus of the protein. And to confirm this, I will look at my second setup where I have deleted the C terminus. So by logic, if I am seeing fluorescence in the first setup, then I should not see fluorescence in the second setup, because after deleting the C terminus, the signal peptide should also get deleted, and vice versa. In case if the signal peptide is present in the at the end terminus, I would not see the fluorescence at the first setup, and but I would see fluorescence in the second setup. So he was very happy after this response. He said, "Yeah, very good, exactly." Then there was a third professor. So he asked me that, Shubham, have you studied glycolysis? So I said, yes, sir, I have studied glycolysis. So he asked me that, do you know what the committed step of the glycolysis is? So first, like I was forgetting the committed step, so by mistakenly, I just said that, sir, it is glucose to glucose 6 phosphate. So he gave me hint like, no, no, it is not the committed step. So he gave me one hint that, you know what a committed step is. So I said, yes, sir, a committed step is a step that after which, like after a product is formed, it cannot go back to it cannot go into any other cycle. It has to complete the process only. So then it suddenly clicked to me. So I said, sir, it should be fructose 6 phosphate to fructose 1,6 phosphate, because because if glucose 6 phosphate is being formed, it can also go to glycogenesis, or it can go to pentose phosphate pathway. But if fructose 1,6 phosphate is being formed, then it cannot go anywhere else. It has to complete the glycolysis. So he was like, yeah, very good. So this was all about my interview experience of MSc Life Sciences at IISc Bangalore, and I was selected for the MSc program, and I got my mail on 27th May. Like the interviews were from 18th to 20th May, and I got my mail on 27th. So after 1 week, I got my mail. So my key insights from this interview would be that you should always be prepared with three topic of interest. So it is not only for IISc, but I was also asked in IISER, TIFRs. I would also share my interview experience at IISER and TIFR in later videos. So, but they usually ask what your topic of interest is. So you should be prepared with topic of interest. Like you should know you should have two to three topic of interest. Another key insight from me would be that you should think loudly. It is not necessary that you know everything. I also did not know everything during my interview, as you have just seen. But what I was doing is I was able to think. I was able to process the questions. I was able to think in my mind, and I was able to put something to the table. So it is much better if you will think about it. If you will if you have some, if you already know some concept, you can connect it to the question which they are asking, and then you can put something, like you can say some answers. It is like not very concerning if you do not know one, two, or three questions. It does not matter that much. But it does matter if you will not try. Like if you will just simply say, "So, sorry, sir, I don't know." So it will not like it will not be fair. So what you should do is you have to be prepared for questions out of the box, and you should be prepared for thinking. Like if you are asked something from out of the box, you should be prepared. Like, yeah, how would I do it? How should I uh how should I think about it? Or how will I do the experiment? Like as they asked me from to set up an experiment, so you should also be able to set up an experiment. So I would suggest that you should know the techniques very well. like uh, like I said that I in my in the cell biology segment, I said about ultra centrifugation, density gradient centrifugation. So you should be well aware with your techniques. You should be well versed. So that is what I would say that you should know very well. And apart from that, I would also say that if you are sitting in an interview, the first thing you have to do is stay calm. It is not necessary that they are not there to reject you. Of course, they are there to select you. So you have to stay calm. If you do not know anything, just wait for 5 to 10 seconds, think about it, and then answer. But it is not necessary like you have to just answer immediately. If you don't know, think about it. Even if you are not able to think something, then you can say, "Sorry, sir, I don't know the answer. Can you please change the question?" It won't matter that much. So that's all for today. I hope you learned something new from this interview experience. In the next video, I will post my integrated PhD interview experience at IISc Bangalore. So until then, happy learning.